In this chapter, we're going to talk about inheritance, but before we even get there, we have to talk about what mitosis and meiosis is. So, when we talk about how this process starts, we kind of have to talk about the two different types of sexual reproduction. There is asexual, which means that one parent will give rise to two offspring, so it's going to be a cloning of itself. Bacteria, algae, fungus can do this. And then sexual reproduction. That's going to be when two individual organisms come together and then mate to create a unique offspring. So one of the things is that cells have to come from pre-existing cells. So in the past, this wasn't something that was commonly known. They used to think that, like, for instance, if you left a blanket in the barn, if you came back a few days later and shook the blanket out, that's where baby rats and mice would come from. So they just assumed that that's how you get mice and rats, where we clearly know that the mice and rats came from, a mom and dad, and then eventually they found a nice, safe place to sleep. If we were to compare sexual to asexual, you can see with sexual, we're going to have individual organisms that look different from the parent. So here's the mom, and you can see that some look kind of close, but others look a little more like dad. They're going to be a combination of genetic material. Whereas if we look at sexual or asexual, they're all going to be exactly the same. So, if you recall back from prokaryotes, the bacteria, they do this thing called binary fission. And that's where one cell will go ahead and split into two. It's definitely asexual because there's only one sort of organism responsible for this. And there's really three steps for them. One is that their genetic material duplicates. The second is that the cell gets longer and starts to move to opposite sides and eventually just splits. So we'll talk way more about this in microbiology, but you could see that here's the cell, here's the genetic material. It goes ahead and starts to elongate. The genetic material is doubling in size. Eventually what's going to happen is that the cell will start to make this little fissure where it's going to start to divide into two and then become two clones of each other. If we look at the prokaryotes and eukaryotes, let's do the prokaryotes first. It's a lot easier to reproduce. You just basically have to find the right space and have enough food, and you can go ahead and reproduce. Because it's so much easier, they can reproduce every 10 to 15 minutes. They reproduce asexually. They don't need to find a partner. They have a small amount of genes. Please don't memorize this number. No nucleus to worry about, so really all they have to do is split in half and move into two cells, and that's it. And their DNA is circular, which makes it even easier to copy. If we look at us, we're a lot more complicated. Even if you look at our cells, we're a lot more complicated right there, but we have to find a partner that's genetically different from us, and then we'll be able to make offspring. Most produce sexually. There are some eukaryotes that can do asexual, so like for instance with yeast. Yeast is a eukaryote, and it can do sexual or asexual. Fungus, because yeast is a type of fungus, they can do sexual or asexual. Humans have about 25 to 27,000 genes. We're still sort of coming up with this number. Please don't memorize this number. But just to give you an idea of how more complicated we are than bacteria. We definitely have a bunch of organelles. The nucleus is definitely there, so it's got to go away when we do this process. And our genetic material is going to be linear, where, again, prokaryotes is going to be circular. So we're really going to concentrate on eukaryotic processes for now on. But eukaryotes basically have a big amount of DNA. Each one of our cells in humans that we can't even see with our naked eye has about 6 feet of genetic information. So if you take that DNA and put it back to back to back to back to back, it'll be 6 feet long. So in one cell, that would be too much, so we have to condense it. And that condensing and coiling process is our next thing that we're going to talk about. You can see that here's that DNA double helix, and if we sort of keep condensing it, eventually it'll become a chromosome. It surrounds itself around these things called histones. These histones will eventually sort of clump up together and then start to even form their own loops, and eventually this is what we have, our chromosome. The chromosome is just going to be tightly packed DNA around these special proteins called histones. Here you're seeing what a chromosome looks like. This is about to double. And what they've done is taken just the tip of the chromosome and exposed it to this chemical that makes it sort of just unwind. And the stringy thing that you're seeing is just regular genetic material. And then these little dots everywhere, those are those histones. We're actually going to go ahead and extract our DNA in a couple weeks, so this will be kind of interesting to see. So chromosomes can be condensed because DNA is so long, but basically what happens is that condensation of those DNA makes it sort of easier for it to organize, and when we do separate the cells out in different phases of mitosis and meiosis, it makes it easier to move them from one cell to another. And if they were really loose and sort of always in that uncoiled sort of look, they would get ripped and torn all the time, and that would be a major problem. So to kind of give you an idea what chromosomes look like, this is what a normal chromosome in your cell looks like. But when it is getting ready to divide, we'll talk about different phases soon. There's going to be an S phase, which is going to be called synthesis phase, and it's basically doubling its genetic material. And it's doubling it because eventually what's going to happen is one of these copies are going to go to a cell, and one of these copies are going to go to another cell. And because of that, we have to copy our genetic material perfectly and move them. So here, I wanted you to see, do you think, number one, we have a copied DNA, or do we have uncopied DNA, and how many chromosomes do we have here? We have 2 chromosomes. They are doubled, which means that they're about to separate. Normally a chromosome would just be 1, but this is the doubled version. Here you can see chromosomes in a normal cell. We've stained them with a special dye which shows us this little black region. These are going to be the usable information of the genetic material, and the whiter regions, they're the non-usable genetic material. And we'll talk about that more in our next unit. But in this little box, you can see that there's 2 chromosomes. You can see that they're different sizes, and you can see that that banding pattern, the white and the black, are a little different. So here we see 2 normal chromosomes. Here we see chromosomes that are about to split and go into 2 cells. So remember how here we had 1 linear chromosome? Here the cell isn't ready to divide, so we don't need a copy. Here we do. So here's a chromosome, and it's made a copy. It sort of has this attachment point where it just kind of keeps itself organized and is able to double check that everything on this side matches everything on this side. And eventually what will happen is these will separate and break, and one will go into one chromosome, or one cell, and one will go into another cell. So you could see that, remember last time we were looking at chromosomes that sort of look like H's? Now we're seeing linear ones. These are normal chromosomes. They're not ready to divide, but they'll be in 2 different cells soon. So when we talk about the cell cycle is made up of a bunch of different parts, and that'll be our next video.